Oh, shoot, my bad.
I just started pretty good.
He didn't even realize his camera was on.
Like, I kind of don't care, but then in the future, if I ever
Yeah.
use this for another class, they're like, who is that guy?
Who doesn't have his shirt on in this recording?
We also have
I'm chilling. I have my pants on. I'm just chilling in my own room. Like, it's not like there's like some clothes requirement for sitting in your own room.
It's okay,
I should let you know, we also have a representative from nursing joining us here today.
How you doing?
I'm like, hi, Hawkins. You know, I sign your time cards.
Yeah. Well, that's good. You already know him.
I do. Totally fine. I have a son, so I understand completely.
Our oldest has been logging into his first class, like lying down in the
Oh,
bed, laptop like this.
Haley did that a few times.
At least he's joining the class.
I mean, I was kind of like, well, we didn't really sign up for virtual, so. But.
Well, you go ahead,
Okay.
and I'm gonna be taking attendance here. Really?
Hi guys, I'm Dr. Amy Edmondson. I am the BSN program coordinator. So I wanted to come talk to you guys about next steps to come into the nursing program if you are interested. How many of you guys are nursing majors? Okay, back half of the room here. Okay, awesome. And I know Hawken is a nursing major. Is there somebody else on?
Yeah. Let me check it here. A student named Alessa Miller.
Okay,
And I think they all have advisors.
perfect. That was going to be my first question. If you don't have an advisor, please let me know. I'm going to give you my card. For any of you guys that are not nursing majors, if you want to make the jump and switch, I'll be happy to talk to you guys about that. You're like, absolutely not.
No, that happens pretty often, actually.
Yeah.
Especially if you've had A&Ps
Mhm.
and lifespan in this class, that's what you need to start nursing, so.
Yeah. So, but I just wanted to talk to you about like your next steps. Are you guys looking to come in for spring 21 or fall 21? Or do you know?
Fall.
Fall? Spring. Fall or spring? Fall. Okay, awesome. So in looking at your trajectory of what you need to do, definitely meet with your advisor this semester. You should be meeting with them every single semester, but it will be important for them to meet with you to look over your science prerequisite grades, your GPA. You do have to have a nursing program. So they're going to look at that with you to ensure that you're on track to maintain that. Also, you will have to take an admission exam to come into the nursing program. Right now, it is our Kaplan admission exam. And currently, we are giving it virtually. So you'll get to sit and look at my face for like 3 hours while you take it on Zoom. So if that's something that you need to do this semester, please let me know. The new dates are on king.edu, and it actually has all of the steps to go through. So you actually have to register through King and also through the Kaplan site. So just remember, it is a 2-step process when you do that. And most people get the King part done and they forget about the Kaplan. If that happens, I'll touch base with you and say, like, this is what you need to do. On that exam, it is not wholly sciences. It has sciences, reading, math, reading comprehension, writing, and critical thinking, all in 91 questions. So I've not taken, you know, how that pans out, but as far as science goes, we do look at science scores and overall scores. You do have to have a 55 on science and 67 overall. So those are things that your advisor will talk to you about whenever you meet with them. Since you are a traditional King student, you don't have to apply through like a separate system. You just need to fill out a petition for acceptance, which you get from us. So you definitely need to let your advisor know when you plan to come in, because I have to send that to you guys, okay? Trying to think about anything else. We do have a Student Nurses Association, so if you all are interested in getting involved in that, right now we're meeting on Teams, but pre-COVID, we had some really good meetings, took our students on conferences, trips to network with other in the field. So that's something you definitely want to get involved with. And we also do a lot of community outreach, so that helps with your service hours too. Any questions for me?
When do we have to take the nursing entrance exam? Like, when is that? When would be a good day to try and take that?
Okay, so that's a good question. Make sure that you are almost completed through ANP2, or that you have completed ANP2. It does have ANP1, it has micro, but it's very heavy on anatomy and phys too. So I usually have an offering every single, and it just depends on like what day it'll fall. But if at the end of the semester I see that some people need to take it that may not have been able to, I can schedule additional offerings. That being said, don't wait until the very last minute. I would take, like if I were an ANP2 now, I would probably take it in November and then in December have a back. You can take it 2 times, okay? We don't want you to have to take it 2 times, and you don't want to have to take it 2 times, but there is that cushion there if you need to have a second try at it. Yes?
So do we have to take it this semester, or can we take it in the?
You can take it in the summer, yeah. I do offerings all through the summer, all the way up until about before classes start. I'll do offerings and wait until that August date to take it. So if you're taking ANP2 in, I would say April, and that way you don't have to worry about it through the summer, and you're all set to.
And also, does it cost anything to take the test?
It is $50, and that purely goes to Kaplan. So we can't change that, unfortunately. But if you do have some need for that fee waived, we can apply for a fee waiver through Kaplan to see if they can help us with that. Any other questions? Okay, I'm going to leave some information with Dr. Ong for you guys. I am in Whitehall 110. I had to think about it. Where am I at? Usually I'm there most days. This week you will not find me really anywhere because we do have accreditation this week. But the best way to get a hold of me is email. I'm usually pretty responsive to email within a couple hours. My cell phone number is on here. If you do text me, just please tell me who you are and how I know you. Hey, I'm Amy, and you came to my micro class today. So that I'll actually, information or hack my phone or something. Okay? All right, guys. Thank you so much for your time.
Thank you for coming.
Oh, I enjoy coming and talking to you guys.
This is my favorite class to teach, and they're a really good group so far, so.
They seem like a really good group.
Yeah, she was gonna come last week, but, anyway, he tested negative. I don't know if I told you that, but.
You
tested
did.
negative,
I had to take
so.
Haley too, because she had an ear infection, and they said that wasn't like
A reason to go back to
And I
school.
said, okay, that's fine, I'll go get
Yeah.
results.
Hey, Hawkins,
Bye, guys.
What up?
could you mute your mic, please?
It was.
Michelle?
I just unmuted it.
Oh, it must be the other person.
Not bad. I will do that now.
Thanks.
There we go. So if you, just to reiterate, if you're in AMP2 this semester, you will have enough of that content by November to take the Kaplan. If you're taking A&P2 in the spring, you should take the April date. You also need to kind of look back through your notes of A&Ps and micro before you take that, because I've had students, like good students, just kind of go take the Kaplan cold and then they have to redo it. You actually need this class and A&P knowledge to do nursing school. So it's not stuff to learn and then forget it. You have to learn it and then really know it. So I want to go back and pick up that one slide of fermentation from last time. And that should be at the end of this slide set. Maybe it's more than one slide. Maybe it's two. It's actually two slides that I care about. This one and this one. So let me make that a little bit bigger. So we last time had been talking about aerobic respiration and anaerobic respiration, and talked about what steps do they have and how are they the same and how are they different. How many ATP do they make per glucose? And fermentation is separate from those two styles of making ATP. So the process of fermentation, let's see if okay. The process of fermentation is different than those other two, because fermentation is going to be making ATP totally without involving an electron transport chain. Fermentation makes ATP without using any electron transport chain. And it is basically doing glycolysis plus a few other steps. There is the beginning, which is glycolysis, but there's not Krebs cycle, there's not citric acid cycle, there is not electron transport chain. It's glycolysis and then some additional steps. The actual point of fermentation, and the reason why cells even do this, is that fermentation is going to regenerate. That's a plus. NAD plus. This right here is NAD plus. It's going to regenerate NAD plus. So why is that important? The other thing that you need to know about fermentation is in what situation do cells need to regenerate NAD plus? Because they actually have an electron transport chain. They actually can do aerobic or anaerobic respiration, but something has made the electron transport chain stop. That's when you do fermentation. So break out of microbe world for a minute and think about when human bodies, when do we do fermentation? What do you say? When we die? Sometimes it's right before we die. That's one time. Do people ever do fermentation? Once in a while. Are you gonna say something?
Are we gonna?
Yeah.
Oh, no.
No? So one of the only times that we do fermentation is if we are exercising so hard that we run out of oxygen. So like think of sprinters, swimming sprinters or running sprinters or bike sprinters. if they're really going hard and you use up all of your oxygen, you actually ferment and produce lactic acid. And you may have heard of that, lactic acid buildup. You had exercise this?
Yeah, I'm in it.
You're in it right now? You've probably not gotten here yet, but we humans can do fermentation and produce lactic acid if our oxygen runs out. And we can't keep on surviving that way. We can't keep on surviving with our oxygen running out, but it's a temporary thing for us. We can keep going a little bit, and we can ferment glucose and produce lactic acid, and then maybe that makes your muscles hurt.
So I have a question.
Uh-huh.
You said fermentation has glycolysis, but doesn't have Krebs cycle or electron.
Or electron transport chain.
But you just said that the electronic transport chain stops.
Yeah, so when the electron transport chain stops for some reason, like running out of oxygen, then the organism will start doing fermentation.
Gotcha.
Let me say that again because that's really important. When the electron transport chain stops for some reason, then the organism, whether it's a person or a bacterium, then they're going to do fermentation at that point, because fermentation is a bad way to make ATP. It's very inefficient. I'm going to erase what's here and keep writing on here, because I'm hoping that this writing shows up on Teams.
Yeah, I can see that one a lot more clearly.
Yeah. I will try to write on here as much as I can without using the sideboard. So on this picture, if you've got this picture, draw up here that the organism would have started with glucose sugar, and during glycolysis turn that into pyruvic acid. So this section that comes before, that's glycolysis. Oh, glycolysis. And that made 2 ATPs and 2 NADHs. Lord. So misaligned. 2 ATPs and 2 NADHs before this. And remember that the NADHs were supposed to go and drop off their electrons They were supposed to go and drop off their electrons to the electron transport chain.
It is not calibrated over there.
It's really not.
I was gonna say, it was doing pretty good on
You know
this
what
side.
happened? I asked IT to come calibrate it. And the guy did, and he's like this tall. So I think he wrote on this part of the board, it all looks good. Yeah, it's good to go. And then I'm trying to write down here, and this is the problem area, so need to ask him to come back.
And do the whole board.
Pretend you're short, then calibrate it. So these are supposed to drop off electrons to the electron transport chain. But if the electron transport chain is not working, NADH has nowhere to put the electrons. There's nowhere to put the electrons. So in that case, all of the NAD plus, all of the NAD plus is getting filled up with electrons and turning into NADH, so it's not available. Or available. It's not available. Pretend that says available. So think about this, like NAD plus is the Uber driver going around and looking for passengers. So all the Uber drivers get passengers, but they can't drop them off. And then at that point, there are no more Uber drivers to pick up people. There are no more places to put electrons. So if this continues, this situation right here, if that continues, it will cause glycolysis to stop. If that continues, glycolysis stops. And then you don't even get your measly 2 ATP. So kind of working backward in this process. Say that in an organism that uses oxygen for its electron transport chain, its aerobic respirator, oxygen runs out. I am going to write this on the sideboard. So I'm writing an aerobic microbe. This is the sequence of events. Oxygen runs out when you're not in oxygen. Next thing that happens is electron transport chain stops. Because you need the oxygen to pick up electrons. So if you don't have oxygen, electron transport chain stops. NADH can't drop off electrons, which means that NAD pluses are not enough in the cell. And then the key question becomes where to put the electrons that NADH is stuck holding. That's what the problem is. So we need NADH to put electrons somewhere if glycolysis is going to continue in this situation. Got to put those electrons somewhere. So what fermentation does is it gives NADH a place to put electrons. This is kind of a simplification, but places where NADH can put electrons during fermentation are into, these are the places electrons can go into. Electrons can go into acids, or alcohols. Acids and alcohols are produced during fermentation as waste products or electron dumps, let's say. Got to get rid of those electrons. They end up going into acids or alcohols, like lactic acid.
What about the other one?
What other ones?
On the quiz, there was one more.
There is one more.
Gotta keep you on your toes.
It's not related to this.
Mhm.
Well, it's kind of related to this. It's kind of related to this. CO2 is also produced during this process. CO2 itself is not a place to put electrons. The other ones are.
Okay.
But all three of these are waste products, and they tend to be produced during fermentation. So, beer, vodka, so on. That's a fermentative product of microbes that are using sugar out of malt for beer or sugar out of potatoes for vodka. It's done in the absence of oxygen to make, we're like making the microbes, starve them of oxygen, make their electron transport chain stop, feed them sugar, and then the waste products become useful to us. They're doing fermentation because they have to, because we're making them ferment. So all the good things like bread, CO2 raises the bread, rises raises the bread, produces bubbles and lightens it. When you make sauerkraut or kimchi, you got the acid going on for fermentation. These are waste products of microbes. So in lab, we do a lot of different tests for what kind of acids can be made, and do we see CO2 coming out, because that tells us whether the microbe's fermenting. So we use that as information to know what kind of microbe are you. Can you ferment glucose? Can you ferment this other sugar that's not glucose? But fermentation is a last resort if you're a microbe. You don't get a lot of ATP out of it, because this is not making any ATP. It's just a place to put electrons from your NADHs filling up. Oh, that's scroll. So this is a really good comparison chart for the 3 styles of making ATP. Divide them like so. And it tells you what are the steps. So aerobic and anaerobic respiration, they've both got an electron transport chain, and that's what makes them respiration. They both start with glycolysis. They both have Krebs cycle. They both have an electron transport chain. It's just that if it's aerobic, they have to have oxygen. They have to have oxygen to run an aerobic respiration chain. And this is the best at making, whoa, that's the best at making ATP. Huh?
But then, so like, if a microbe is anaerobic, then does it technically not have to do fermentation because oxygen is not already in its system?
Yes. If it's, okay, it's a yes if. Hold that thought. So an aerobic respirator, in the absence of oxygen, it will ferment. An anaerobic respirator will ferment if it doesn't have its electron acceptor. Anaerobic respirators, if they've got their electron acceptor, like their version of oxygen, they would rather do this than this because the ATP difference is so great.
Yeah.
2 ATPs is just so pitiful as far as powering your. Let me finish writing on here. Their electron acceptors. So take, for example, E. coli. E. coli. E. coli is a facultative anaerobe. It has a lot of options. So if E. coli is in oxygen, it will be an aerobic respirator and it will make maximum ATP. If there's no oxygen, it can do anaerobic respiration as long as it has sulfate. So it's like, okay, I don't have oxygen. If I've got sulfate, I can do this. If I don't have oxygen or sulfate, then it would ferment. If it had to. If it had to. So that's kind of related to your question talking about, well, if they're anaerobic, why would they ever ferment? They have to, if they don't have their electron acceptor and their electron transport chain stops, then they would ferment.
Okay, that makes sense.
Good question. So this chart is telling you a lot of the differences. And right here on this chart, I want you to write something in the space, and I will attempt to write it in the space, which is helpful. So it's got this pink box and it says fermentation using organic compounds as electron acceptor. You've got alcohol or acids produced as wastes. And they are the place where those electrons are put. Put those put. Pitiful. Those are the places where electrons are put as waste. And these tend to characterize fermentation. And we also have CO2 produced in all three of these.
Electron puts
Put.
waste into protein.
Electrons are put into these and they're waste products. Yeah.
And then what dictates whether the microbe will make alcohol or acid, or does it make like equal portions of both and CO2?
Depends on the microbe. So it depends on what enzymes they have, and some of them will make multiple acids and alcohol.
Mm-hmm.
Will only make one acid and alcohol. And the details of that I do not enjoy.
Okay.
I tried to teach it once in the majors class, and it's a lot of chemistry that I just didn't care about. But there's something called a mixed acid fermentation where you make multiple kinds of acid. But people who do homebrew and are really into that, or people who go into brewing industry, they would know a lot about that. And there are tricks that you can push you get a certain strain for your beer that makes more alcohol and less acid. That'll make it taste different. If you got a mixed acid strain for fermenting your beer, it'll make it taste different. But the details of that, I'm not very strong in. These two slides, you can cross off. This one and this one, I'm not going to talk about those this year.
As if it never existed.
This is the very first one we turned on.
Okay. So we'll kind of move on to a new topic, but it's not totally a new topic. It's still related to metabolism, because the style of metabolism that a microbe runs, whether it's anaerobic or aerobic or fermentation, it's going to depend on what kind of enzymes does that microbe have, and the enzymes depend on its genetics. We are not anaerobic respirators because we don't have the proteins in our electron transport to do that, because we don't have the genes in our genome to let us do that. So anything that a microbe does that's cool, it's got to have the genes to allow it. That's how this topic kind of relates. So to review, in our cells, we keep our DNA in a nucleus, and we are dipped, which means that we have two copies of each, basically, one from each parent. Humans do not have plasmids. There are some fungi and protozoan organisms that have these extra DNAs called plasmids, but we don't. Viral genomes, I'll talk about that when we get to viruses, but bacteria, they are keeping their DNA in a circular chromosome. They are haploid, which means that they have one copy of each gene. And they may also have some extra helpful genes on their plasmids. They may be carrying around some genes that are helpful but not essential on these extra DNAs called plasmids. And whatever genes they've got are going to determine what this microbe can do. So the genes are going to determine the abilities of this cell. It doesn't so much matter whether the genes are on the chromosome or the plasmid, but if you've got a gene, that gives you the possibility to do something, to do a behavior or a possibility or a new metabolism.
When like a bacteria divides, does it also copies its plasmids then too? Because then how would the next generation have the same plasmids?
It does. So before a bacterium would divide, it copies its chromosome, and the plasmids also copy. They have ways to ensure that both daughter cells get one chromosome. The plasmids are just kind of like when it pinches together, some happen to go in one and some happen to go. They do copy, and they do get transmitted to the offspring cells. So whatever microbe's going to do, and this is E. coli. This is a picture of all of the DNA in an E. coli cell spilling out. It's really long. It's all one big DNA. The genes give it the abilities because the genes give it proteins. So the proteins would include enzymes, and structural components of this bacterium. Like E. coli can make a flagellum. That at least looks good. There we go. E. coli can make a flagellum, and the flagellum is made out of protein. So microbes that make a flagellum, they have to have a gene to tell it how to make the protein in this tail. We don't make flagella because we don't have the gene to do it. That would be stupid for us. Like, what would we do? Don't. It would be too small to move us. But any protein that gets made, there has to be a gene to encode that. So bacteria still have DNA genomes. That's similar to us. And it's still a double helix. And they still use DNA for their genome and RNA for their transcripts. But what I want you to do in this space are what are the DNA bases and what are their complements? What are they matched with? What?
You said, say it one more time, please.
Yeah, what are the DNA bases? What are they?
A, G.
Yeah. A, G, C, T. So those are the types of DNA bases.
Are we
And
gonna
what,
know the names of them or just like the letters?
in this class, you don't have to know the spelled out names.
Sweet.
Just, okay. What do they match with? What's the match of A? T. G matches with C. C matches with G. T matches with A. It's A. T matches with A. And those matches come from shapes and hydrogen bonding being complementary between those two chemicals. So the structure of DNA and the way that DNA is used in bacteria is the same as in us. That's not a big difference. They just only have one copy of each gene, and they keep their genes on a circular chromosome. This part is the same. And remember that DNA being, having this complementary base pairing means that you can separate a piece of DNA in half and use each half to build in the other half. So it allows for easy copying. It allows for conservation of genetic information in the daughter cells. All of that's the same as in people. Faithful copying. Like if you have unfaithful copying, what that would mean is that your offspring wouldn't get the right gene sequence.
So does that lead to like, genetic defects?
Sometimes. Yeah. I think that's worth putting on here. If you don't have faithful copying, that's going to lead to some type of mutation, which may be good or bad or neither. But the whole structure of DNA is set up to make it likely that you will have faithful copying, and the next generation of Staphylococcus will end up with the same genes as the parents did, or the previous generation. Parents is not a good word for microbes. But you have the same genetics being passed down through each generation of cells. So the genes are important, and the proteins are important to let the microbe do something. There's a step in between that is transcription. So you may have learned this in high school, called the central dogma. The central dogma of biology is that we keep our genetic info, let's say our stable genetic information in DNA, and we have proteins that are doing the work of the cell. It says work. Proteins are doing the work of the cell. The proteins get information to be constructed originally from DNA, but we have this intermediate. There is an intermediate between DNA and protein that is RNA. So we have to transcribe RNA, which might be one of these three versions, and then translate that information into a protein. One thing I want you to know off of this picture, this process is transcription. And this process is done by an enzyme called RNA polymerase. RNA polymerase is the enzyme that reads DNA and makes an RNA copy of it. The process of going from RNA to protein is called translation. And that job is done by ribosomes, like so, ribosomes. Just having the gene in your genome is not good enough unless you actually use the information to make RNAs or to make proteins. Just because you've got a gene doesn't mean that you're using it. You usually have to make protein or be making some specific RNAs to be using that information. And the reason why I teach you to this, teach this to you, sorry, teach this to you, is because if you mess up or destroy ribosomes, you destroy the life of a microbe. If you mess up the action of ribosomes, you will destroy the life of a microbe. This is for people too, but we do it really often to microbes. So bacterial ribosomes are disrupted, by many antibiotic drugs. Add on the word bacterial. Bacterial ribosomes are disrupted or targeted or messed up by a lot of different drugs that we use as antibiotics. Let me think of one. So, think of one that's specifically for ribosomes. If you've ever had an infection and you took tetracycline or doxycycline or one of those type of drugs, that's killing bacteria by messing up their ribosomes so that they can't make protein. This is so essential that if you take away this ability, you kill the microbe. Here's what's useful about this. Our ribosomes are slightly different shaped. They're slightly different in shape. They work basically the same. And they have the same big overall mushroom-looking shape, but our ribosomes are different enough from bacterial ones that we can take these drugs, and the drugs mess up these, but not our ribosomes. That's excellent. That's just excellent news. So a lot of the antibiotics that gets prescribed for bacterial infections are messing up ribosomes so that the microbe then cannot make proteins and then it will die. That's the main reason to care about this in a molecular microbe or medical microbiology type class. So this next slide goes with this process. So this next slide is about translation. Translation. Okay, good. Translation. So what this is showing you is specific sets of 3 RNA letters. 3 RNA letters. So these are representing RNA, and these words are representing amino acids. So remember that proteins are made of linked, folded chains of amino acids. And I want to go back a picture and show an example of that. So what the ribosome is doing is it is reading the messenger RNA sequence here, and it's reading this in a set of 3 RNA bases, and each 3 bases will give an amino acid that goes into the protein.
How come,
Uh-huh.
how come we're not going over the transcription of DNA?
Because I don't have time in this class.
Oh, okay.
That's a really good question. Brooke said, why aren't we going over transcription? So I don't have time in this class. It is very important, but because we do a lot of ribosome targeting for drugs, this is where I focus in this class. So to get a protein that works, you have to have the correct 3D shape, and you have to have the correct amino acids in that chain if the protein is going to work well. So what the ribosomes do that is pretty genius is they will link up a certain 3 letters like CCC and convert that into a proline amino acid in the protein. So you can see here the names of the amino acids. You don't have to learn those in this class. But I want to draw your attention to some certain triplets that do something that's not making an amino acid. So you see there's one triplet that tells start making protein. It's called the start codon. And there are certain triplets that tell the ribosome stop making protein. These are called stop codons. So each one of these triplets is called a codon. Codon. C-O-D-O-N. There we go. Codon. And this is the key to how DNA information gets turned into protein information. So as ribosomes are doing this process, they have to have these linking molecules. We have these linking molecules that link up DNA codons, or sorry, RNA codons with specific amino acids. And the molecules that do this are called tRNAs, transfer RNAs. So each one of these transfer RNAs will come to the messenger RNA, and it has a complementary match. G with C, C with G, A with U instead of T. RNA has a U base instead of a T. And on this end, the tRNA would be carrying threonine amino acid. Threonine. So they're kind of the key to decoding. They have one end that matches the codon of the messenger RNA. On the other end, they're carrying the amino acid. And as this process is working, it's converting DNA information into protein information. I think I want to stop right there for today. Stop there for today. And for Wednesday, go ahead and get ready for Wednesday for the next topic, which is viruses. I'm not going to skip viral material this year. We may do a little bit of what's left in microbial genetics, or we may just go straight on to viruses. But go ahead and do your quiz for viruses, reading for viruses, because they're really important this year and every year.
And then, Dr. Ong, what do you want me to do for lab tomorrow?
So, yeah, I'm glad you asked that. Lab tomorrow is a makeup. So if you don't have, if you're not missing any experiments that you still need to do, you don't need to come to lab tomorrow. But there's 3 of you that do have labs that you need to do, or part of a lab that you need to do. So if you're one of those 3 people, come to lab tomorrow at your normal time. But otherwise, we're not having a new lab tomorrow. And this is also because the materials did not come in, because Becca and I both thought the other person ordered it a month ago. Nobody ordered it until Friday. So now I don't have the stuff. So we're going to push back a week in lab. Or if you want to come, go ahead.
So then I'm also going to miss like next week because I get out on the 28th, I believe. And so then I'm just going to have to do like an extra long lab to make up for what I've missed, or just schedule a different time where I can come in and catch up.
If things go well and you get out on the 28th, that's a Monday.
As I say, yeah, you would be at lab on Tuesday.
Yeah, you would be able to be at lab Tuesday. So that should be okay.
Thank you.
Thanks for asking. Okay, I will see some of you in lab tomorrow. If you want to come, like, see me tomorrow afternoon, if you have questions about stuff, go ahead and do that. I will be here and available. I just don't have the materials to do the new lab until Thursday. Oh, yeah. I've also got Dr. Edmison's info here, and course plan and business cards and so on. Yeah.